Hello everyone, my name is Robert. Welcome to my channel. In my next videos, I will present the observations under the microscope, biological and non-biological things. In today's video, I will tell you about onion cells. Here is all the things that I used. A microscope, tweezers, a slide, a cover slip, a pipette, a red or yellow onion, water, and blue food coloring if you are experimenting with a yellow onion. Tear off a layer of peel with tweezers, as thin as possible. Then place it on a slide. Add a drop of water. Then put the cover slip. Let's put it under the microscope. Then look from smallest zoom to largest. On this photo, we can see the nucleus, which contains all the genetic information of the living creature. The vacuole is a bag of water that can shrink during drought and contains cyanidin and anthocyanin, leads to dye onions red. The wall and the plasma membrane are fused. The cytoplasm is a bag enclosed by the membrane and the wall. I also have the onion specimen, which was colored by the blue dye. We also see the nucleus, the vacuole, the cytoplasm, and the plasma membrane, which was fused with the wall. If you like my video, then subscribe to my channel, click on the bell, put it for all. Thank you for listening. I hope you enjoy it. To the next video.